Okay, so tertiary structures are the actual three-dimensional structures that amino acids will take on as they are joined by the ribosome. So as they form the peptide bonds, the side chains will interact with each other to form these tertiary structures, and they'll form a certain shape. Quaternary structures are just multiple, usually two or more, tertiary structures that come together to form certain proteins. Now, all proteins will have quaternary structure, but most of them, if not all of them, will have tertiary structures to them. All right, so going into metabolism, just go on. I'm not even gonna do a joke. I'm not going to do a joke.
No joke this time. We weren't spontaneous enough for that. I get it. You weren't spontaneous to do it. Oh, that makes sense.
Yeah, exactly. No joke this time. So the overview for metabolism is that the word itself refers to all of the chemical reactions within an organism. Includes catabolic activity and anabolic activity, so the breaking of larger molecules into smaller and the building of smaller into larger. There must be some degree of negative feedback for metabolism. It has to be able to be told when to stop breaking down larger molecules or when to stop building them up. Glycolysis, the Krebs cycle, and photosynthesis are probably the most commonly talked about and well known to the general public and also undergraduate students that refer to a certain type of metabolism. They generally obey a few specific rules. One of those is that catabolic pathways, so the breaking down of a certain molecule, cannot have the same pathway that an anabolic metabolism pathway would have. So the breaking down of glucose in glycolysis cannot share the same pathway of something trying to build up glucose. And then they have to occur in specific parts of the cell or organism. For glycolysis, it happens in the mitochondrial lumen. For the Krebs cycle, it happens outside of it, and then photosynthesis takes place both in and outside of the chloroplast, thyroid membranes, et cetera, et cetera. So they're very specific in where they happen. Yeah. Okay, so going over glycolysis real quick, this is a very general, slightly detailed mapping of it. I mean, it's general because it really doesn't go into everything, but one of the other rules that metabolism has to obey is that there has to be a first committed step. So in terms of glycolysis, when glucose gets captured by hexokinase, it will phosphorylate the glucose out of its ring shape so that it can further be broken down at certain parts in the glycolysis metabolism. This first step with hexokinase is the committed step. So once this happens, it's not going to go back to the ring shape unless something physically acts upon the glucose 6-phosphate that results from that reaction. And if that were the case, it would not involve hexokinase because that pathway cannot be used to build back something. It can't go in the reverse direction. So once that happens, that basically kicks off glycolysis. It'll follow through these 10 general steps until glycolysis produces pyruvate, which will eventually go into the Krebs cycle, or well known as the citric acid cycle, which pyruvate will be taken up by acetyl-CoA, which is an acetyl group attached to a coenzyme A, and that'll pull pyruvate into the citric acid cycle, where it basically just gets recycled to form precursors for high energy donation of some form. So NAD plus becomes NADH2 and some protons, which have high energy for breaking and building bonds, which will also facilitate the formation of ATP from glycolysis. Photosynthesis, not going to go too in depth with this, but just know there's a light reaction which uses light to power an electron transport chain through different chlorophyll pigments. Energy is given off by the breaking of water, which also gives off protons and OHs, which end up reducing into O2 gas, which is why they help us breathe. And then the light independent reaction is just what helps generate those precursor molecules for generating energy, which will take place at night in light of not having any light. And also just kind of help those precursors be there for the light reaction in general, for the formation of energy. So genetics, it's a poor fellow we blame everything on. So like, oh no, my skin, must be my genetics. Specifically for the biochemistry portion of this, we're going to look at the genetics of protein expression. So where proteins come from, how those end up getting translated and transcribed, and then the eventual expression of that protein, since those are what's most commonly looked at in biochemistry. So it starts off in the nucleus, where RNA polymerase binds to a sequence of DNA at an initial starting point. This is translation, and the end result is an mRNA molecule that will eventually leave the nucleus into the cytoplasm of the cell, where the rest of these different processes will end up taking place. Before it can leave, though, it gets a 5' cap and a poly A tail, which is done by different polymerases, or not polymerases, but different enzymes. The poly A tail is done by a different polymerase because it adds on multiple adenosines, but the cap is a different enzyme completely. Once that mRNA gets sent out into the cytoplasm with its 5' cap and poly A tail, it is going to be captured by a ribosome, specifically the small subunit. Once it's captured by the small subunit, a acetyl tRNA, which is just a tRNA that has its amino acid on it for methionine, which is basically the starting amino acid. It'll bind to that captured mRNA, and once that binds and that complex says, hey, we have the start codon, we're good to go for transcription, the large subunit for the ribosome will come, and the initial position for that starting codon will be in the P site. Once that's there, multiple tRNAs that have their amino group on them will come down into the A site. Peptide bonds will form between the amino acids, and the ribosome will move down the mRNA template with different tRNAs that have the anticodons to those codons coming in, binding the amino acids together to form that amino acid chain. And as it moves down, they fall out of the E site, which is basically just the exit site. So once it's bonded, it's empty, no longer needs to be there, so it falls out and gets repaired with an amino acid. These chains that are made by transcription are the primary and secondary structures of the proteins, and the tertiary structure is not actually complete until the amino acid chain is completed. If there's multiple sections to that tertiary structure from the secondary, it's not completed until those come together, and then those tertiary structures may come together to form a quaternary structure for that protein. Once the protein is completely formed, whether it be a tertiary or a quaternary structure, the protein will go into expression somewhere in the body or the cell or whatever. Ribozymes are responsible for the peptide bond catalytic activity that the ribosomes do, so that's, or do execute, which is a really cool form of a protein working to make other proteins. Kind of funny there. And then real quick, just in case it might show up on the exit exam, I don't think it will, but the genetic pathway for just DNA in general, when it replicates, DNA is attacked by DNA helicase, specifically at least two enzymes of it. They unwind DNA, so there's a string of DNA here, one will bind here, one will bind there, and they'll go like that, and basically unwind it into a giant loop. DNA polymerase will bind after a primer is set at an initiation point, and that'll start to code the complementary pair to one of the strands of DNA. There's leading and lagging strands. The lagging strand is the side that can't be fully coded from 3' to 5', and it results in different fragments called Okazaki fragments, which are ligated by a DNA ligase after everything's pretty much said and done. Those will get fused with the rest of the strand, so everything's nice and good to go. Other factors that contribute to the proper replication of DNA are DNA gyrases and topoisomerases, which are generally the same thing. DNA gyrases are usually found in bacteria though, or at least called that. What they both do is prevent supercoiling while DNA helicase unwinds it. So further down, if you were to take something that was corkscrewed and try to unwind it, the ends that are corkscrewed will try to supercoil on themselves. So topoisomerases or DNA gyrases help alleviate that stress so it doesn't supercoil and end up destroying the DNA farther down the line. Cool. So we're good. Cool. Bam. Good luck. You do well.